Ig class switching. First of all, I explain the structure of immunoglobulins. Immunoglobulins have two light chains and two heavy chains, and they have two regions, variable region and constant region. Ig cross switching is the mechanism by which B cells, in response to an external stimulus, change the constant region of the antibody they produce by DNA recombination to a class appropriate for the site where the antibody is required. Activated by antigen binding, B cells proliferate immunoglobulins of the IgM and IgD types, but by antigen stimulation or the action of cytokines released by helper T cells, they produce antibodies of the IgG, IgA, and IgE types. In this class switch, the constant region where the antibody binds to immune cells and complement changes, while the variable region where it binds to the antigen remains unchanged. This results in the production of antibodies with different functions against a particular antigen, allowing more efficient elimination of the antigen. Next, I explain how class switching is occurred. First, when RNA polymerase II is transcribing the S region, the FACT complex generates specific chemical modification on nucleosomes in the S region to mark them for DNA cleavage enzyme. Second, the expression of a protein called AID activates DNA cleavage enzyme. Third, cut out a particular area of the constant region. Fourth, remove the cut DNA fragment and join the remaining parts together.
Why class switch is occurred? Human beings have 5 kinds of antibody. We call them S-type. At first, activated B cells can produce only IgM and IgD. However, thanks to class switching, they can produce other kinds of antibody. For example, IgG can neutralize a toxin, enable leukocyte or macrophage to find the pathogen, and so on. It plays an important role in immune system. Without cross switching, we can't gain IgG and get sick easily.